Hello, my name is Cameron Burke and welcome to this Kaplan MCAT Science Review for membrane components. This video covers all the most important parts of the cell membrane, including lipids, proteins, carbohydrates, membrane receptors, and cell-to-cell junctions, all of which help each other out and make sure cells work properly. The main goal for this review is to remember the major parts of the cell membrane and what they do to keep the cell working the way it's supposed to. At the end, you'll get to practice these learning goals on questions like this one about hemidesmosomes, so keep that in mind as you go through the video. The core of the cell membrane is composed mostly of fatty acids, which are basically just a long greasy carbon tail with a carboxylic group stuck on the end. There are a couple different types of fatty acids. The most basic type is saturated, where every carbon in the tail has as many hydrogens as it can hold, and then it's called a saturated fatty acid. Sometimes there aren't as many hydrogens attached as there's potential for. If there are double or triple bonds instead of hydrogens, it's an unsaturated fatty acid. Saturated fatty acids have different properties than unsaturated fatty acids. If fatty acids are unsaturated, the double bonds create a kink in the chain, which makes it harder for the acids to stack up on each other in a uniform way. If a membrane has more saturated fat, it's less fluid because the fatty acids can stack up. And adding unsaturated fat will make it more fluid because the kinks disrupt stacking. Understanding fatty acids is important because the majority of a cell membrane is made up of lipids. When a cell membrane is viewed as a whole, it's thought of as a fluid mosaic model because all the parts are a little different in size and shape and they're connected through chemical bonds, but not super crazy tight. This results in a group of molecules kind of floating around together in the same general space with everybody doing their part to keep the cell going. However, if you zoom in and look closer, you can see the phospholipids making the separate bilayers of the membrane, with the hydrophilic phosphate group heads making up the majority of the outer surface of the cell, and the hydrophobic fatty acid tails making up the inside of the bilayer. Fatty acids play an important role in storing energy, usually by being conformed into triglyceride molecules with 3 fatty acids attached to a sugar backbone. When fatty acids are incorporated into cell membranes, they're changed into glycerophospholipids by replacing the third fatty acid with a phosphate group, so the glycerol backbone has 2 fatty acids and a negatively charged phosphate. This allows part of the molecule to be able to interact with water instead of being repelled by it, so both a hydrophobic and a hydrophilic region of the bilayer can form. Technically, it's called a glycerophospholipid, but on test day, it will probably just be called a phospholipid. While lipids as a category are the major component of any cell membrane, there are actually several types of lipids you can expect to find in the bilayer, each with its own specific job. The first special category of lipids is the sphingolipids. Sphingolipids are atypical because they're created from sphingosine, which is a large amino alcohol. There are 4 types of sphingolipids you need to be able to recognize: ceramide, sphingomyelin, cerebrosides, and gangliosides. The second category that you need to care about is cholesterol. Cholesterol stabilizes the membrane in extreme heat and gives it fluidity in extreme cold, and it also helps out with steroid synthesis. Finally, our third category of special fat is waxes, which is a long fatty acid chain with a long chain ester. The wax's job is to give stability to the hydrophobic region of the membrane. Waxes are pretty rare in mammals. They're mostly found in plants. Together, these different lipids and other molecules present in the membrane create a fluid mosaic model. There are 3 types of proteins that join lipids to make up the cell membrane. Integral proteins account for 2 of these 3. Proteins that have anything to do with the interior of the membrane get counted as integral. The third type is peripheral, or membrane-associated, which are proteins that just kick it on the outer edges of the membrane, but they don't actually go inside the cell's membrane. Transmembrane proteins usually include proteins who have jobs to do on both sides of the cell membrane, so both on the inside of the cell and on the outside of the cell. This includes but isn't limited to transport proteins, receptors, and channels for molecules to get through. Some examples of transmembrane proteins that you might want to know about include aquaporins, which let water through, ion channels, which let things like chlorine through, and the sodium-potassium pump, which regulates sodium and potassium and is extremely important to know about on the membrane because it's such a cross-topical subject. Embedded proteins can associate either with the interior or exterior of a cell membrane, but not both. 2 examples of embedded proteins are prostaglandin synthesis 1 and 2, which are the proteins responsible for forming the lipids that function like hormones to cause smooth muscle contraction, which are located in the outer bilayer. Peripheral proteins generally hang out just on the outside of the cell and can bind to the membrane and to other transmembrane proteins or to embedded proteins. Some important examples are cytochrome C and cholesterol oxidases. But cytochrome C is extra special because it's vital for ATP production, so that's a good protein to know when you take the MCAT. While proteins do most of the heavy lifting in terms of moving molecules around, carbohydrates within the cell membrane play a role in sending and receiving chemical messages. They tend to be mostly hydrophilic because they have so many oxygen molecules, so they're on the outside of the cell, which allows proteins to attach to them. Often carbohydrates are the signals the immune system relies on when looking for infection, and they're also what identifies cells pathogens could potentially target. Carbohydrates on the surface of cells can also allow for the formation of biofilms, which is when a bunch of cells, typically bacteria, like pictured here, all hang on to each other to more effectively stay on a surface. This is what happens when you have bacterial plaques on your teeth. One example of carbs impacting cell identification is differing blood types. The various carbohydrates on the outer side of the membrane of blood cells is what catches the attention of the immune system if someone is transfused with an incompatible type of blood. While they can be used for identifying cells, receptors on the membrane surface can also turn on or turn off transporters needed for facilitated diffusion or active transport. Usually these membrane receptors actually cross the entire membrane. The receptors can either just go ahead and open the channel or participate in biosignaling that will eventually result in the channel being opened. One example of this is the ligand-gated ion channel. The receptor opens a channel in response to a specific ligand, coming in and binding. So the ligand is present, and the channel lets in molecules. Some examples of such ligands are dopamine and acetylcholine, which couldn't function as neurotransmitters without receptors in the cell membrane helping them. Intercellular junctions are an element of the cell membrane that allows cells to stick together to form one layer, to pass chemical messages or molecules back and forth, and to get molecules into the extracellular matrix. On the MCAT, there are 3 types of intercellular junctions to worry about: gap junctions, desmosomes, and tight junctions. Each of the 3 junction types have their own special jobs, and their structure is usually a pretty good clue about what that job is. Gap junctions are little tubes that allow for direct communication. They're made up of 6 little connects-ins that team up to make one big connects-on. They can open or close to let in water or other molecules from one cell to another, or from the cell to the extracellular space. Chemical communication between cells is important, but cells also need to be in physical contact. That's where desmosomes come in. A desmosome's main job is to bind adjacent cells by grabbing onto the next cell's cytoskeleton. Desmosomes are what is holding on all your epithelial tissue, particularly in your skin, together. So basically that's pretty important. They work by tying together the filaments from 2 transmembrane proteins on 2 adjacent cells. Hemidesmosomes are special examples of desmosomes that are basically just half of a regular desmosome. Instead of tying 2 cells together, they anchor one cell into the basement layer of a separate tissue. This part is what holds your skin onto your muscles, so your skin doesn't fall off your body even if it's attached to itself. Again, that is a pretty important function. It's important that sheets of cells don't fall off your body, but it's also important that all the ions and plasma stay inside the cells themselves. Tight junctions keep epithelial cells close together so nothing can leak out from between them. They form a tight physical chain on the outermost layer of cells in the epithelium. Tight junctions are responsible for creating a transepithelial voltage difference between the lumen and the interstitium. Together, tight junctions act like a giant rubber band that stops fluids and ions from leaking out of the space in between cells. Now you have the chance to check out how your learning goal progress is coming along. The first practice question tests your knowledge of cholesterol. The correct answer is D. Cholesterol has nothing to do with differentiating saturated fatty acids from unsaturated fatty acids. The second practice question addresses the difference between transmembrane and peripheral proteins. The correct answer is D. Peripheral proteins strictly hang out on the outside of the cell membrane, while transmembrane and embedded proteins hang out within the cell membrane. The final practice question covers hemidesmosomes. C is the correct answer. Hemidesmosomes attach cells to basement membranes and other tissues. To recap what you need to know about lipids, unsaturated fatty acids increase fluidity. Saturated fats decrease fluidity. Phospholipids form the phospholipid bilayer and have one less fatty acid than triacylglycerol. Sphingolipids are lipids derived from sphingosine. Cholesterol plays a role in stabilizing fluidity in the membrane, and waxes increase rigidity in plant cells. The components of the cell membrane include proteins, carbohydrates, membrane receptors, and cell-to-cell junctions. Protein types include transmembrane, embedded, and peripheral. Carbohydrates are important for forming biofilms and for cell identification and messaging. Membrane receptors help control ion flow, and cell-to-cell junctions include gap junctions, desmosomes, and tight junctions, and all are important for cells' abilities to work together. Thank you so much for watching this Kaplan MCAT Science review about membrane components. We hope it helps you out on test day.